So Ronithe Senatus is a first-generation immigrant from Haiti. She moved to the US during the summer of 2012, and she earned her Bachelor's of Science in Neuroscience and Behavior in 2018 from Florida Atlantic University in Boca Raton, Florida. She is currently a second-year research track student in Dr. Asma Zaidi's lab. She's planning on attending medical school and hopefully specializing in neurology, but is keeping an open mind. So the floor is yours.
Can everyone hear me? Okay, so as Anne-Marie just mentioned, my name is Ronithe Senatus, and the title of this presentation is Neuroprotective Effects of Polyunsaturated Fatty Acid, PUFA, on a neuronal calcium transporters. So here is an outline of the different section of my presentation. A little bit of background. Neurons use pulses of calcium to help regulate major signaling that control critical processes such as gene expression, learning and memory, synaptic plasticity, and neurotransmission. There is a 10,000-fold gradient of calcium across the plasma membrane, with the inside being the lowest. There are many systems in the cell working to maintain that gradient. But the most important one is the plasma membrane calcium ATPase, PMCA. It is a 10 transmembrane calcium pump. It uses ATP to pump calcium out of the cells. And previous study in our lab has shown that PMCA protein and activity tend to decline with age, in neurodegenerative disease, and under condition of oxidative stress. When we lose those PMCA, it leads to an increase in intracellular calcium, which disrupts many major functions and eventually leads to death. So this is where my project comes in. The goal of my research is to identify intervention that would boost the PMCA activity and prevent neuronal cell death. Currently, there has been a lot of research and a growing interest in polyunsaturated fatty acids, also known as PUFAs. So PUFAs are healthy fat. They contain multiple double bonds, and the most important ones are omega-3 and omega-6 fatty acid, and some examples are DHA and EPA. They are localized in the plasma membrane, and they help regulate important membrane proteins such as transporters, receptors, and channel. So here is a demonstration of how those PUFAs work. Here on the left, we have the plasma membrane with only saturated fatty acid. It is very viscous, and these fats tend to be solid at room temperature. On the right now, we have the plasma membrane with unsaturated fatty acid. They introduce kink, which make the membrane more fluid. And these fats tend to be liquid at room temperature. One of the PUFAs that we'll be focusing on is docosahexaenoic acid, DHA. It is an omega-3 fatty acid. It contains 6 double bonds, with the last double bond located 3 carbons away from the metal end. DHA has been shown to have many beneficial effects, and I listed a few here, such as reduction in cancer, cardiovascular disease, neuroinflammation. It helps promote healthy aging. It has antioxidant property. However, there hasn't been a lot of studies investigating the role of DHA in calcium homeostasis, which brings me to my research question. Can DHA boost PMCA activity? So we hypothesize that exposure of our cells to DHA will boost PMCA activity and protein level. First, let me introduce you to the cells that we use. They are the SHSY5Y cell line. They are neuroblastoma cells. They are adherent, which means that they attach to the plate that they are cultured in. And many other studies have used them to study neurodegeneration, neurogenesis, and neuroprotection. These cells can be differentiated to be more neuronal-like using retinoic acid for treatment of 7 days, which make them the appropriate model for my studies. So on panel A at day 0, we have the SH cell when I just plated them. They are round and they look like regular cells. And on panel B, after being exposed to retinoic acid for a day, you can see that they start to grow neurite projections. And on day 6, on panel C, you can see that those neurites are fully grown and they start to form network. So to investigate our hypothesis, we did the following experimental strategy. First, the cells were differentiated with retinoic acid. Then they were exposed to DHA at different concentration for 24 hours. Then we measured cell viability. Then we determined PMCA activity. We determined the PMCA protein level, and finally measure intracellular calcium. Now on to the fun part, which is my result. First, we looked at the effect of DHA on cell viability. Basically, we wanted to see how much DHA can the cell take before they start dying. So on this graph, on the x-axis, we have DHA concentration in micromolar, which will stay consistent throughout the rest of this presentation. And here on the y-axis, we have cell viability in percentage. And overall, our results show that DHA preserves cell viability up to about 200 micromolar, where we start experiencing some level of cell death. Then we looked at the effects of DHA on the PMCA activity. Now on the y-axis, we have PMCA activity expressed in nanomoles of phosphate over milligrams of protein over time in minutes. Our results show that there was an increase in PMCA activity, a dose-dependent increase, with the most being at about 50 micromolar, which was about threefold. So since PMCA activity increased, we wanted to see what was happening to the PMCA protein level. So we did some immunoblotting. On the left, we have the PMCA protein on our Western blot, which is about 140 kilodalton. And I use beta-actin as the loading control. And then we use ImageJ to do a densitometry analysis. Now on the y-axis, we have the band density that we measured. It was interesting. We only saw a dramatic increase in the PMCA protein at about 100 micromolar of DHA. So then we went on, since the PMCA protein wasn't increasing as the PMCA activity, we then on and investigate the functionality of the PMCA itself, which means that if we have increase in PMCA, we should have less calcium. So we plated the cells, then we exposed them, we treated them with fluorophore, which is a dye that penetrates the plasma membrane, get to the cytosol, and binds to calcium. Once it binds to calcium, it creates a green fluorescence. So our results show, using flow cytometry, show that there was a dose-dependent decrease in intracellular calcium, which correlates with the increase in PMCA activity. So to conclude, DHA did not alter cell viability. It caused a dose-dependent increase in PMCA activity. It caused a small increase in the PMCA protein, and it led to a significant reduction in intracellular calcium. So we have increase in PMCA activity, but not a drastic increase in the protein level. So we had to understand what exactly is DHA is doing to the cell. So we came up with two ideas. So we think that first, DHA could have an indirect effect on the PMCA protein. It could be modifying the membrane and making the environment more likable for the PMCA, which make it more active. Or it could have an indirect interaction. It could have a direct interaction with the protein itself and making it more active. So we have a way of actually investigating that. We have a method in our lab where we can actually isolate the PMCA and remove it from the membrane. So we could then expose it to the DHA. So if we observe the same increase in activity, then we could determine exactly where that DHA is making its move. So for my future direction, we would like to evaluate the mechanism underlying the DHA-mediated increase in PMCA activity. Then we would like to assess the effects of other omega-3 and omega-6 fatty acid or a combination of both to see if the effect is generic. And lastly, we would like to investigate the potential protective effects of DHA on PMCA activity and protein level when it is exposed to ROS. So I would like to thank the following people, specifically Dr. Zaidi, my committee member, anyone associated with the College of Bioscience, my amazing lab mate and RA, and my cat. Thank you. Any questions?
Hi, Ronithe, great job. I just had a question about why, just basic, like, why did you pick those cells and why did you decide to differentiate them?
At first, I did the experiments with differentiated cells, undifferentiated cell, I should say, and I did observe an increase in PMCA. So we wanted to take our experiment a little bit further and make it look more like what we would see in the brain by differentiating them and make them more neuron